Okay, so metabotropic receptors, unlike ionotropic receptors, do not have a channel, within their own structure. However, as I showed you previously, the G protein released from a metabotropic receptor upon binding of a neurotransmitter can actually move along the membrane and interact with a channel in such a way that it opens a pore to allow for depolarization or hyperpolarization, allow for ions to pass through, right, after a delay. This is another example of what these G proteins can do, and this example should illustrate the power of these metabotropic receptors in terms of responding to the message of the neurotransmitter being released by the presynaptic cell. So here again is our postsynaptic membrane, okay? There's the neurotransmitter at the top, those little stars that were released, right, presynaptically into the cleft upon the arrival of an action potential. This is a blow-up of the membrane again. So we have our, phospholipid bilayer, and then we have our metabotropic receptor. And I show here a neurotransmitter bound, right? The structure has changed its shape in response. A G protein has been released, and this time the G protein travels along the membrane and interacts with some other complex here. Anytime you see a big box or a triangular shape here, it's a protein, right? Coded for by your genetic material and inserted into the membrane, somewhere in the cell. Now, the binding of this G protein to this complex, right, results in a change in shape, right? And this activates what we refer to as second messengers. So think of it this way. The G protein released inside is the first messenger, right? This is telling the cell that there's activity. Something has happened out here. The previous neuron is firing, is releasing neurotransmitter that's, diffusing across the cleft. This is the first messenger. This is now interacted, right? It's been released and now interacted with something else, and it's activated these second messengers, okay? Now these second messengers, I want you to understand, will actually activate, third messengers and fourth messengers and fifth messengers, and then downstream, referred to as downstream of this activation, some of these chemicals will actually enter the nucleus of the postsynaptic cell, right? And when they get there, they will actually bind to areas of the DNA. They will unzip certain areas. They will promote transcription and then translation of new proteins. They will, in other cases, bind and close certain regions of the genetic material, and they will turn off transcription and translation of certain genes. So by utilizing the message, right, the activity that is present in the synaptic cleft, that information about how much activity is occurring, how much neurotransmitter is being released, is being conveyed through this chemical communication system all the way back to the DNA. And it's going to result in a different looking postsynaptic cell. Some of the proteins that may be turned on might be different sorts of receptors. So that in the future, this postsynaptic membrane will respond differently to the neurotransmitter that is released by the presynaptic cell, right? It could maybe produce fewer receptors, so there's less of a postsynaptic response. It may produce more, so there's a heightened postsynaptic response. It may produce a different set of receptors. You can start to get a sense about how drugs, which are chemicals, which will act at many of these receptors, many of the metabotropic receptors, will stimulate in an unnatural fashion, some of these postsynaptic membranes. And that will result in changes in gene expression and changes in protein synthesis, changes in the actual physical structure of the postsynaptic cell. There can actually be literal growth of new connections in response to activity, right? There can also be retraction in response to other forms of activity. So this hopefully illustrates the power of the metabotropic receptor to interpret the chemical message being received, right, in the synaptic cleft, and communicating that information via the G protein, the first messenger, via the second messengers, via these biochemical cascades, right, all the way back to the genetic material which will result in physical structural changes to the postsynaptic cell.